Excellent. Well, good morning, everyone. Welcome. Bienvenue tout le monde en Ni, Kinawea, and from British Columbia Smithers, Danaweye. Welcome to the FishCAST seminar series. This is our last one of our Spring-Summer Series 2022, and we're so excited that you were able to join us. We've got such an amazing seminar speaker lined up for you today. I normally I'm calling in or I'm talking to you from Kingsville, Windsor, Essex region in southwestern Ontario. But today I'm actually coming in to you from Smithers, BC. And this is Smithers, BC is located on the unceded territory of the Gidimt'en clan and the Wet'suwet'en people. And I'm just so honored and privileged to be able to just enjoy this incredible land and the incredible sockeye salmon in Babine Lake. I'm just really excited to be allowed to share the bounty that I see in front of me, and the bounties that you see in front of you, and the privilege that we have in working on the lands and the treaties and the unceded territories. What's FishCAST? So for those of you who are just joining us for the first time, FishCAST is an NSERC Create funded graduate training program, and it's in freshwater fisheries and fishes conservation and management. It's basically, I think, still one of the only freshwater fisheries directed training program for graduate students. And it's made up of principal investigators from 6 different universities in Manitoba, Quebec, and Ontario. We are so happy we're just finishing up our second year of a 6-year-long program. We already have over 25, 26 trainees in our program. And we're really trying to build a community. We're trying to build a community with our partners. And so we're so happy to have practitioners and partners on our seminar series as well as involved in our FishCAST community. We have over 50 partners from government, NGOs, industry, Indigenous communities, and just conservation authorities, and we see some of them today. And together and collectively, our communities, we're as you can see, we're trying to move beyond land acknowledgements like the one I just gave and transition more into meaningful partnerships and relationship building, where we try to really acknowledge the history, the connection to and the knowledge of the land on which we're situated. But more so than that, collectively, it's just been amazing how through our seminar series, through the partnerships we're curating, through the co-development that we're doing, our micro-credentials, we're really trying to address systemic barriers in our fields and try to work collaboratively amongst all of us to try to find ways and working with folks like you to overcome them. We're all treaty land inhabitants. We're all working together for equality, inclusivity, and diversity. And this is just one of the ways we try to do that. Part of our training, like you see today, is by inviting researchers and practitioners to speak to our community about the work that they do, the experiences they have, and to share their knowledge of the issues that are facing our Canadian fishes and fisheries. The where we can, I'm so pleased to be able to highlight what's researcher today, and that's Dr. Valerie Berseth. Dr. Valerie Berseth, she's an environmental sociologist, and she works in the area of climate adaptation, conservation, and natural resource management. Dr. Berseth, she completed her PhD in sociology at UBC, the University of British Columbia, and she completed her Master's of Arts, MA, in Socio-Legal Studies at York University. That's amazing. But what does Dr. Berseth actually do? Well, she investigates the meaning and the value of wildness, which is a really interesting concept, and the ways that people understand and manage the risks of human interventions into nature with a particular focus on genomic science and technology. So an amazing way to integrate and to weave together so many different disparate disciplines and fields and technologies. She's currently working at the University of Carleton University with Dr. Vivian Nguyen, who's one of our partners, and she's also involved in FishCAST. She also works, Dr. Berseth works with Dr. Stephen Schott at Carleton, Dr. Nicole Klenk at the University of Toronto, and she's focusing on right now on the risk perception in forestry and how that's related to the spread of mountain pine beetle. But to do so, and how it's the use of genomic selection to try to manage pests and how it responds to climate change, the overall goal of improving forest health. And so basically, it's not such a, forestry is the focus, salmon are the focus, but it's really this concept of wildness and the human footprint and the use of technologies and trying to address these wicked problems in conservation. And so today we are so pleased we're going to be hearing about Dr. Berseth's work on what is it that makes a salmon wild. So in essence, examining how salmon hatcheries deal with the challenges of artificial reproduction and how genomic science is changing and what it means to conserve wild fish. And so without further ado, I welcome you to help me welcome Dr. Berseth this morning and giving us her amazing seminar that we are so anxious and happy to hear. So thanks so much, everybody.
Thank you so much for that very wonderful introduction, and I really appreciate the way that you sort of worded the large goals that I'm trying to tackle in this work. I'm going to go ahead and start sharing my screen. Okay. So thank you very much to Kendra, Christina, and the rest of the team at FishCAST for organizing what is a really fantastic series of talks. I've watched many of the previous ones with great interest, and I've learned a lot. And I want to thank everyone for coming today. Today, I am going to be sharing some of my dissertation research on Pacific salmon and this question of wildness. And as Christina mentioned, I'm a sociologist by training. And one of the things that I try and do in my work is to bridge some of the gaps that exist between sociology and the social sciences with natural science disciplines that might be focused on some very similar issues or the same issues, but from different lenses. So what that means is that often I find myself in rooms of salmon biologists or genomicists who look at me and ask, what is a sociologist doing here? And I get a similar question when I'm in rooms with a bunch of sociologists who ask me, why are you studying fish? But so much of how we manage the environment is about managing people with different values, different knowledges, and different goals. So I'm really grateful for spaces like FishCAST where I can come and connect with all of you and talk about some of the really important intersections of these different worlds. And I'm not an expert in genomics. I'm also not an expert in fish biology. So I'm always grateful for any sort of opportunities to learn from people in these fields. And especially, I know there are practitioners here today, there are graduate students. I'm very keen to connect with all of you. So starting off with a brief land acknowledgement. So the work that I will be discussing today was conducted on the traditional, ancestral, and unceded territories of the New Channel, Musqueam, Tsleil-Waututh, Squamish, Stolo, and Secwepemc peoples. And I'm also calling in today as a settler living in Kenyan-Kayaka territory. And as Christina mentioned, land acknowledgements are a very small and can often be a superficial part of what are foundational obligations that we have to the land and people in these places to conduct our research in a good way. And so it's part of this continually evolving learning process in my life. So in my work, I look at this practice of artificial reproduction. Humans artificially breed millions of plants and animals every year and release or relocate them into the natural environment. And this is done for several reasons, including to provide harvest opportunities for hunting, fishing, and forestry, as well as for conservation objectives to rebuild or sustain struggling wild populations. And one of the key questions for these programs is whether the plants and animals that they produce can be understood, classified, and made sufficiently wild to survive and coexist in the natural environment. And this idea of humans actively producing wildlife presents an interesting paradox. In Western culture, the idea of a wild place or a wild organism has conventionally been understood as nature that is untouched by humans. But in order to deal with accelerating biodiversity loss, wildlife conservation typically requires more and more human intervention. And there's been very little research into the social dimensions of artificial reproduction and how people in these programs understand the meaning of wildness in this context. So in my work, I look at artificial breeding programs as fruitful sites to explore how people and organizations wrestle with human interventions into nature, and importantly, how they understand and manage the risks of these interventions. So today I'm going to talk about two studies that I conducted during my PhD as part of a larger project called EPIC4 that was funded by Genome Canada, Genome BC, and Genome Quebec. And so the first study looks within the hatchery system and asks how hatcheries produce wildness in Pacific salmon. And the second study looks at the potential use of genomic information to selectively breed salmon with the idea that hatcheries could potentially produce salmon that are more resilient to climate change. So first, just a brief background on Pacific salmon hatcheries, though I'm sure that there are many of you who are more familiar with this case study. So for decades, Canada and many other countries have been artificially breeding Pacific salmon in salmon hatcheries. And hatcheries are facilities where workers take sperm and eggs from mature salmon, mix them together, incubate them, and rear them until they are old enough to be released into the natural environment. And we do this at a fairly large scale, although globally, Canada represents a small portion of Pacific salmon hatchery production. So there are 23 hatcheries in BC that release over 300 million Pacific salmon per year. And in order for hatcheries to achieve these dual goals of conservation and harvest opportunities, they need to be able to substitute naturally spawning wild salmon with salmon that are grown in hatcheries. So this is related to an idea that Foster and colleagues call substitutability, or the idea that environmental limits are surmountable because of our ability to substitute things in nature. But research on salmon genetics has shown that hatchery salmon have some differences than naturally spawning salmon. And in some cases, this is poor survival rates, lower reproductive success, and other physical and behavioral differences that affect survival. And some studies have found that these changes can occur within just one generation of hatchery production. So over time, the accumulation of scientific research on these differences between hatchery-born salmon and naturally spawning salmon has prompted a debate among people who ask, can hatchery salmon still be considered wild? So what I wanted to do in this study was to examine how hatcheries deal with this problem of substitutability. How do they produce salmon that are as close to wild as possible? And what role does genomic science play in this process? So being a sociologist, I come at this with a particular theoretical framework, and that in this case is biopolitics. So biopolitics is a field of research that stems from the work of Michel Foucault, a French sociologist, who argued that in modern society, power is exercised in ways that value and support some forms of life over others. And this is done by controlling various biological processes like birth and reproduction. So in other words, not all life is valued equally. And if we want to understand how life is managed, we need to look at the knowledge and techniques that are used to manage humans and non-humans as well. So typically when people started doing biopolitical research on non-humans, they were looking at domesticated animals like livestock in agricultural settings. But over time, there's been a growing focus on how biopolitics plays out in situations where humans are involved in monitoring, regulating, and intervening in the lives of wild animals. And some research has looked at the value that's placed on wildness as a characteristic in these settings. So the data for this research come from 46 interviews with Fisheries and Oceans Canada employees. So they came from different areas of DFO. There were 6 genomic scientists, 13 people working in the salmon enhancement program at the management level, and then 27 staff working at 15 different hatchery facilities. And the analysis focused on how participants define wildness, what knowledge and practices are used in hatcheries and in the salmon enhancement program in order to produce wildlife, and what forms of life are targeted for intervention. So I found that while some respondents defined wildness in the conventional sense of salmon that have not been altered by humans, they also viewed wildness as a function of genetics. And according to hatchery staff and managers, genomic science has had an impact on the way that salmon hatcheries operate because it allows them to translate this concept of wildness into practice. Genomic science can be used to distinguish between wild and domesticated life. And this allows hatcheries to adjust their practices in order to reshape salmon to be more genetically wild. As a result, wildness is understood and practiced differently depending on the scale that you look at. So I'm going to go through each of these scales. So starting out at the very basic level, if we're thinking about wildness as related to genetics, what is a wild gene? There were 2 ways that respondents interpreted this. The first is in terms of lineage. So wild genes are genes that are passed on from wild parents and grandparents. And the second is in terms of natural selection. So wild genes are genes that allow salmon to survive in the natural environment. And these 2 ideas of wild as genetic inform some of the decisions that get made at other levels. So moving up from an individual gene, now we're looking at individual salmon. So what is a wild salmon? In Canada, the wild salmon policy defines a wild salmon as one that is 2 generations removed from the hatchery. But if hatchery fish and wild fish swim in the same river, how can you tell them apart? There are 2 ways that this is done in practice. One is that hatcheries will remove the adipose fin, which can allow fishers to target only hatchery fish for harvest, while letting wild salmon return to spawn the next generation. But as some of you may be aware, not all hatcheries in Canada mark their salmon with removing the adipose fin. So another method for telling if an individual is wild is by analyzing its DNA. This way, hatcheries could screen out hatchery fish and use only wild salmon in breeding practices. So moving up from the individual scale, the next level asks, how wild is this fish in this particular environment? Epigenetics is a branch of genomic science that looks at how your environment changes the way that your genes are expressed. Some studies have shown that hatchery environments produce salmon that are less fit to survive in the wild. So some hatcheries are experimenting with making their rearing tanks look more like natural salmon spawning habitat, which you can see on the right. And they're doing this in different ways by introducing rocks or branches into the tanks. Sometimes there's an employee who will walk above the salmon with a plastic fish to kind of scare them. But this push to alter salmon genetic expression towards something that's more wild isn't coming from a top-down science policy. It's driven by some hatcheries where particular managers and staff are concerned about the salmon that they're producing. And they may not have formal training in genomic science or epigenomics, but they are evolving their practices in response to new genomic knowledge, and combining it with some of their own ideas and views of wildness and ethics for how to care for the animals that they produce. So now moving beyond an individual fish, we're looking at wildness at this level in terms of families and lineages at the broodstock level. A tool that's been introduced but not yet widely adopted in hatcheries is parentage-based tagging. So using genomic science, some hatcheries are starting to map out salmon family trees. And this can allow hatchery staff to select males and females that will produce more genetically wild families. And it's not currently being used for this, but some hatchery workers, when we asked about, what are the potential benefits of genomics, saw parentage-based tagging as a way to mimic what they think is natural mating behavior of salmon. So this hatchery worker, for example, had a particular emphasis on the fact that in nature, they think that it's more natural for a large fish to seek out another large partner. And so there was some desire to use parentage-based tagging to more closely mimic that practice. In some cases of extreme conservation concern, this might mean parentage-based tagging is used sometimes to check fish that come back to the hatchery. And if a fish is not part of the genetically unique stock that the hatchery is trying to conserve, those fish are culled. And this practice has produced some disagreements or tensions among staff about how ethical this practice is. So for example, this technician described ethical issues with killing fish that from their view don't need to be killed. They see it as more natural for salmon to mix across rivers than it is for humans to try and intervene and make some sort of ideal strain of fish. So looking at the population level, the question here is, how wild is a particular river? Respondents talked about a lot of different risks that hatchery fish can pose to wild populations, including overcrowding and consuming limited amounts of food available. So a new method for measuring the wildness of a river is using PNI, or proportionate natural influence. And this involves calculating the percentage of hatchery fish in a river and classifying it according to this chart from very wild to very hatchery influenced. And this tool provides a stream-by-stream assessment of wildness and can be used in developing annual plans for hatchery production to alter the number of fish that are produced, when hatcheries should release salmon, and into what particular rivers or creeks. And lastly, at the level of salmon species, the focus here is on wildness in terms of overall hatchery contributions to the system. So to increase the overall wildness of Pacific salmon, this might mean closing hatcheries or drastically reducing production. And these decisions are based on several factors, including the delicate balance of economic and conservation interests. However, hatcheries also have enormous public support, and several respondents described the subject of closing hatcheries as something that is taboo in fisheries and oceans. And as this hatchery biologist describes, until recently, it's not something that people would even feel comfortable mentioning. Now they explain that they're starting to be able to talk about it, but they still see it as an unlikely prospect. So this shows that there are limits to how far people within and outside the hatchery system are willing to go in this pursuit of wildness. So what I found through looking at Pacific salmon hatcheries is that the meaning of wild has changed. So we don't just think about wildness in terms of a pure, pristine, untouched space or organism. The term wild has expanded to include this idea that wildness is a function of genetics. And it's not just a shift in our understanding. This view of genetic wildness is being translated into practice as hatcheries are adapting to reshape salmon to be more genetically wild. And genomic science provides tools that can allow hatcheries to distinguish between wild and domesticated forms of life and to encourage the proliferation of wildness at multiple levels from genes to species. And they can also weed out certain forms of domesticated life or target them for harvest or culling. And at the same time, I found that this pursuit of wildness is uneven. So I found that there was greater ability to experiment within hatcheries and less flexibility to pursue wildness at higher institutional levels, as well as some different ideas and ethical concerns about how far we should be going in order to produce wild salmon. So this underscores some of the social, political, and economic difficulties in addressing environmental problems. While some of these smaller changes in hatchery practices can shift salmon towards something that's more wild, there are some aspects of this issue that can't be solved through technological advancement alone. And I think a good example of this is this idea that in some spaces it's even difficult to have a conversation about making potentially big changes. And that those might conflict with other values or institutional norms. So for the second study, I wanted to investigate the potential use of genomics to inform breeding practices. And I came to this interested in this question of whether hatcheries should selectively breed salmon, in particular for climate resilience. So, it's possible to use genomic sequencing to identify traits like disease resistance or tolerance of warmer waters. So I asked two questions in this study: should hatcheries selectively breed salmon? And how do people weigh the risks of intervention against other risks like climate change? So some previous literature on this has suggested that with the onset of this idea of the Anthropocene, that we've shifted towards being a more risk-taking society. So in other words, we're more open to actively shaping or altering nature. But there is also some literature on why people tend to distrust or oppose altering nature, including perceptions that certain technologies are unnatural, a distrust in science, or if there is high amounts of uncertainty. But a lot of this research that exists has primarily focused on the general public and surveys that ask about should we alter nature in very abstract terms. And there's a need for research on how people weigh these risks in applied contexts. So for this study, I conducted 105 interviews with people involved in different areas of salmon management, harvest, and stewardship. And I asked, should hatcheries selectively breed salmon for particular characteristics? And why or why not? If so, for what characteristics? So the responses that I got fell into three different categories that I've plotted here along a continuum from less to more risk tolerance. You'll note that they don't add up to 100% because interviewees often expressed more than one type of risk response. So for example, they might oppose selective breeding in some cases, but are open to it in others. I found that most people expressed some opposition to selective breeding, and fewer expressed support for producing salmon that are climate resilient. So I've termed this here risk taking. So calling back to that idea that was raised in the literature that we might be more open to shaping nature. What I didn't expect going into this research was that a significant proportion of respondents were interested in selective breeding for managing the risks of how we already intervene. So I've termed that here risk management. Among those that expressed an aversion to risk, there were several reasons given. Some were rooted in social and cultural beliefs that intervening intentionally in salmon breeding is unnatural. And a lot of people gave the example of this is playing God. Others were concerned about material risks, such as the loss of genetic diversity that, in their view, is key for salmon survival in changing environments. For people who gave a risk management response, they started from the position that humans have already altered salmon from a wild state, and selective breeding can help address some of the risks that we know that hatcheries pose to wild salmon. So some saw selective breeding as a way to improve genetic diversity and address this issue of homogenization. And others wanted to address the fact that hatchery salmon tend to be smaller than naturally spawning salmon, although there were concerns that this might lead to simply producing large fish to support trophy fishing. And lastly, people who expressed support for selective breeding generally argued that climate change has made this level of intervention necessary for salmon survival. So I didn't find anyone in this category who universally supports altering nature for whatever purpose. But when weighed against the risks that climate change and other threats pose, they saw selective breeding as an acceptable response. However, again, there were different visions of what this would look like. Some supported selective breeding specifically for traits that we know will be important for new climate realities. Others envisioned producing salmon that are generally robust. So looking for what is it that makes salmon overall more able to survive any threat. So rather than a debate between two camps, those that support and those that oppose selective breeding, I found that respondents weighed different types of risk in their responses. This included socially constructed risks and ideas about what is natural or unnatural, and material concerns about the consequences of human intervention, like domestication. And part of what determined people's responses was which aspect of wildness they prioritized. So, for example, people who saw selective breeding as a way to correct past mistakes and rewild salmon were more concerned about salmon being wild in terms of their genetic properties than whether they had been, at some point in their history, altered by humans. But still, I found a lack of consensus on what characteristics should be selected for. And ultimately, this research demonstrates the need to examine how people perceive the risks of new technologies in applied contexts. So a couple of concluding points here. So this research provides some new insights into the changing meaning of wildness. I found that through advances in genomic science, wildness is now understood both as this kind of conventional understanding of something that has not been altered by humans, but also in terms of its genetic makeup. And understanding that these two dimensions exist simultaneously helps us to better understand how people weigh different types of interventions and threats to salmon survival. Second, I found that there are multiple levels at which hatcheries are altering salmon or have the ability to alter salmon in this pursuit of wildness. As I mentioned, this process is uneven, and there's little consensus, again, on when and how these interventions should occur, as well as the ethics of doing so. One of the limitations of this work is that I focus primarily on genomic science use in the context of Canadian settler environmental practices. But it's also important to note that Indigenous ways of knowing and managing salmon are an essential part of this picture. And First Nations hatcheries are implementing genomic knowledge in their hatcheries alongside traditional knowledge in innovative and important ways that were outside the scope of the research that I was doing. And lastly, I just want to sort of articulate that this work demonstrates some of the needs to bring together sociology and genomic science in fisheries management. I can speak to the sociology side of things. A lot of the research, the limited research that exists on genomics, has focused on medical applications. And I think that there are a lot of valuable contributions that can be made by bringing together some attention to the social processes and contextual factors alongside some of the biological and ecological dimensions. Some of the implications of this work. First, for science communication, some previous research has shown that scientists tend to avoid engaging with people that they think will oppose their work. But what I found in this research was that conversations about genomic technologies were not simply for or against. A lot of the conversation was when and how to use genomics. And people are not, I think this work kind of challenges the deficit model of the public. I found that people without genomic training are actively seeking out and aware of genomic science publications. In terms of policy responses, I think that this work demonstrates the need to consider social and institutional risks and barriers in developing plans like climate adaptation plans. And I found a lot of support for policies and approaches that are slow, experimental, and work from a ground up approach rather than top-down policies. And lastly, there's a lot of future research opportunities here, including how these risk perceptions change over time or in response to climate events like the heat dome we saw last year. Different types of interventions, as well as different settings. So does it matter if we're talking about salmon or if we're talking about trees? So that's some of my current research. Okay, so that's the end of my talk. I just want to thank my research participants, funders, and collaborators. And I'm very excited to talk with you all.
Thank you so much, Dr. Berseth, for that incredibly interesting and really, really relevant talk to our community as well. So I really appreciate it. That, and if those folks in the audience could also join me in thanking Valerie again. I did see some claps going on, so that was really, really great. We'll stop recording.